So, ladies and gentlemen, this is Learned here, and we're going to attempt to finish the remainder of our note packet on antibacterial medications. So, this picture that you've got is depicting the different ways in which antibacterial drugs function. What is their actual target within the cell? So I want you to take a look at these 5 things. Here's the deal though. Yes, you do need to know the 5 indicated sites of action. You need to know what are the 5 ways that antibiotics actually work. You do not need to know the specific antibiotics listed in the figure. Okay? You do not need to know the specific antibiotics from the figure. So when we're looking at the different targets, cell wall synthesis. Hey, Morgan. And so cell wall synthesis. This is something that we talked about at the beginning of the school year. We talked about how important that peptidoglycan in the cell wall was. And so in terms of cell wall synthesis, things like the beta-lactum drugs, vancomycin and bacitracin, all impact synthesis of the cell wall. Peptidoglycan cannot be put together and laid in those sheets. So would you consider that bacteriostatic or bactericidal? Remember what the difference is between those two. Would you consider the action of those drugs on cell wall synthesis? If you take one of those drugs like vancomycin, it actually prevents the bacteria from creating new cell walls. Is that going to maintain the population of bacteria, or is it going to and prevent them from reproducing and creating more, or is that going to kill them?
Where are we?
On the page that says targets of antibacterial drugs, and then this image is up there. If I go back a slide, so it looks like that. Evan? Okay. What do you think? Bacteriostatic or bactericidal? Static. Yeah? So bacteriostatic, remember, means that it doesn't kill the cells that are alive, it only prevents them from reproducing. Well, is peptidoglycan in the cell wall being produced when the cell is living, if it's already in existence? Or is peptidoglycan only being made when the cell is reproducing? That's kind of the question you have to ask yourself. So now let's look at nucleic acid synthesis. Things like the rifamycins and the fluoroquinolones impact the production of new DNA and RNA. Bacteriostatic, bactericidal.
So what's silent?
Kills them. Think homicidal, and that'll help. That'll help you remember that bactericidal kills any cell. Doesn't matter if it's reproducing or not. Static, think about the growth curve. When the growth curve becomes static, it's stationary, and so it's only impacting reproduction. It's not killing the cells. The growth curve isn't going down. How about cell membrane integrity? What does that mean? Cell membrane integrity. Like the strength of the cell membrane, right? If that's compromised, if the cell membrane gets compromised, is the cell going to survive? So is that going to be, Ailish said no. Is that going to be bacteriostatic or bactericidal?
Bactericidal.
That one's cidal. Okay. Metabolic pathways. So impacting the metabolism of the cell. Static or cidal? Cidal. Yep, I would agree. And then protein synthesis.
Static. No? Silent.
Does protein synthesis only occur in a cell when it's reproducing, or is it happening all the time?
All the time.
Yep.
It's both.
It's probably going to be cidal. It's probably going to kill any cell, whether it's reproducing or not. Ailish first.
So wait, nucleic acid synthesis is bactericidal or static?
Static. It's only making new DNA when it's reproducing. Sarah, was that your question?
Yeah.
Yep. Okay. All right. So, in some cases, the drugs that are used actually have ribosomal targets. And so it's not just one of the 5 structures of the cell, but inside at the ribosome, there are targets that can occur. So these, I'm not super concerned with you knowing. I wanted you to be familiar with them, so that's why I included them in the note packet. But you do not have to know these 7 items for the test. Okay? You only have to know the 5 cell targets, not ribosomal targets. So if I'm looking at the ribosomal targets, some drugs will block the initiation of translation and cause the misreading of messenger RNA. Okay? If messenger RNA is being misread, is that going to be cidal or static?
Silent.
Cidal, because that has to do with protein synthesis. Tetracyclines block the attachment of transfer RNA to the ribosome. Again, cidal, good, because that's going to impact protein synthesis again. Streptogramins, yes, streptogramins, each interferes with a distinct step of protein synthesis. Protein synthesis? Probably cidal again. The macrolides prevent the continuation of protein synthesis. Static or cidal? Cidal. Chloramphenicol prevents peptide bonds from being formed, so the resulting protein can't hook together. Cidal again. Preventing the continuation of protein synthesis. Are you catching on that there's a pattern here? Interfering with the initiation of protein synthesis. Cidal again. So all of the ribosomal targets, if you take any of those medications, those are bactericidal. Now again, if you're taking any of these drugs listed on the screen, think about it. Are these going to be selective in terms of their therapeutic action, or are they going to be non-selective? Non. So that means they could impact any bacterial cells' protein synthesis, not just those that you're infected by. Okay? Pretty important to make that distinction. All right. So let's talk about these beta-lactam drugs. The beta-lactam drugs irreversibly inhibit enzymes involved in cell wall synthesis. So these drugs have a shared chemical structure, and that's how they got their name. But the beta-lactam drugs include penicillins, cephalosporins, monobactams, and carbapenems. The reason I wanted to pull this out for you and show this diagram to you again, do you remember looking at this picture at the beginning of the year when we studied the bacterial cell wall structure?
Mm-hmm.
So it's the same thing, and now taking a look at this, the beta-lactam drugs interfere with the formation of the peptide side chains between adjacent strands of peptidoglycan by inhibiting the penicillin binding proteins. So penicillin is what is actually going in and interfering with that cell wall synthesis. It's a perfect example. Penicillin is one of the examples I do want you to know for the test. Penicillin is a beta-lactam drug, and it targets cell wall synthesis. Okay? That's one you should know. You may not know this, but tuberculosis is one of those diseases that's considered re-emerging at this point. And I'm using that term. It's one of the terms that we're going to study in the future this semester. But it's an infection that there used to be a high incidence rate of. Then it kind of went away, and now it's coming back again. And part of that is because of the development of resistant strains of Mycobacterium tuberculinum. So there's a limited range of antimicrobials that can actually be used to treat Mycobacterium, and that's due to several different factors. This species of bacteria has very slow growth, and their cell wall happens to be really impervious to a lot of drugs. So because they reproduce slowly, it's hard for us to use any of those drugs that are considered bacteriostatic. So a group of 5 medications called the first-line drugs are preferred as they are the most effective as well as the least toxic. They include ethambutol, isoniazid, pyrazinamide, rifampin, and streptomycin. They're commonly referred to as peers. So you get diagnosed with tuberculosis and you immediately go on all 5 of these medications daily. So you're on peers. This is the first line. In some cases, people have been, they've taken peers, the infection appears to go away, and then 6 months to a year later, it comes back. That's how slow reproducing the bacteria that causes tuberculosis actually is. So here you might be able to destroy many of those cells, but then it just takes that long in order for those few cells that were resistant to reproduce and for there to be enough to actually cause the infection again. Go ahead, Alex.
What is tuberculosis again?
So tuberculosis is a lower respiratory tract infection. The tuberculosis bacteria get into your lungs and they kind of create like a colony inside there. They actually become encapsulated, so they create these capsules of bacteria that just continue to grow and reproduce inside your lungs. In some cases, if the infection gets big enough, you actually have to have tissue, like literally lung tissue, cut out.
And it just makes it hard to breathe. Or...
Yep. So you end up sounding like you've got pneumonia, like a severe cough. You can't breathe. You get really tired. High fevers all the time. Amanda?
Isn't there like a TB shot though?
There is, and that one's preventative.
So it works?
The preventative does work, yes. Yep. So as like a school district employee, when I got hired, long time ago, right, well, I had to have the skin test done first. And as long as I tested negative to the skin test that I didn't have tuberculosis, then I was able to get the TB shot, yes, as a preventative. So the peers medications, generally an individual is given a combination of 2 or more. If they're concerned that the individual has been exposed to already resistant strains, a lot of times they will go on all 5, but most of the time it's usually a combination of 2 or more. So that kind of helps prevent the development of resistant mutants, because remember, the more medications you take, the more likely that you're not going to have something that's resistant to every single one of those medications. All right. So, how do we determine if a microbe is even susceptible to an antimicrobial drug? First of all, there's what's known, there's 2 different terms that we're going to look at. One is called the MIC, M-I-C, and the other one is called the MBC. These are going to be the minimum inhibitory concentrations and the minimum bactericidal concentrations. So these are quantitative tests. You can actually collect data, collect numbers, and determine the lowest concentration of a specific drug that prevents the growth of an organism in vitro. Okay? This concentration is called the minimum inhibitory concentration. What is the minimum amount that I can get, or yes, amount I'll say, or concentration that I can give this drug that it actually prevents the growth of an organism, stops them from growing. So the fact that an organism's growth is inhibited by a given concentration of a drug doesn't necessarily mean that an infection can be successfully treated with the drug, right? Inhibited means static, means the bacteria are still there. You're just preventing them from making more and making the infection worse. Where cidal means you're killing all of those cells, entirely getting rid of the infection. So that minimum bactericidal concentration is the lowest concentration of a specific antimicrobial drug that kills 99.9% of a given strain of a bacteria. Go ahead, Alex.
So for the static one, that you still experience symptoms if you have a static.
Yep, but hopefully it's repressing the population of bacteria enough that your own immune system then can fight it off. That's the idea anyways behind it.
Okay.
So the minimum bactericidal concentration, the MBC, is determined by assaying for live organisms in those tubes from the MIC test that showed no growth. So you take a tube of bacteria in a broth where you have a known concentration of bacteria, so you know how many cells are there. You do the minimum inhibitory test, and so you've got bacteria that are still present in the broth because they weren't killed, they were just maintained in terms of their population numbers. You take those same bacteria then and test them against, I just lost my train of thought. You test them by adding in the antimicrobial, the drug again, and test varying different antimicrobials to determine which one most successfully destroys all the cells in that culture. Okay? The MIC and the MBC are important to know in the pharmaceutical field. If you're trying to sell a drug to a health system or to a doctor and say, this one's a really good one, because they do go in, they do presentations for doctors and have to explain, why is this drug better over another drug? Why should you prescribe this one? And so by being able to tell them it's got low toxicity, it's if you can use these terms, this is the type of thing that's going to convince people to buy your drug. So this is one of those things where you might not ever use this stuff in the future, but you never know. You just never know. All right, so this is what the MIC determination looks like. And I want you to look at the screen because I'm going to ask you to interpret this data. Interpret these results. So let me describe what's going on. Remember that a broth tube, you can see the broth, right? And it's usually light in color. And when bacteria begin to grow in it, what does it look like to you? What term would you use to describe it? Kind of foggy looking, cloudy, maybe opaque, where you can't really see through the broth anymore. That term is called turbid. Turbid. So,
I thought that was like when something's really hard.
not with the growth of bacteria. So you are looking for turbidity in the broth, the cloudiness. Okay? So we're looking at organism A. Okay? The first tube is a control. There is no bacteria. In the second tube, they've added 16 micrograms per milliliter of broth of the antimicrobial. Okay, whatever that drug is. That's the concentration of the drug. 16 micrograms is a lot. 8 micrograms is still a lot. 4 micrograms, still a lot. Is there growth in 16, 8, 4, 2, 1? What is the lowest concentration that you can see growth in? 0.5 for organism A.
6.
It's 0.6. It's 0.06. 0.06. So what would be the minimum inhibitory concentration of the drug that would prevent the growth of bacteria? 0.12. Now I know that the answer's right there. You can see it on there, right? But now look at organism B. What's the minimum inhibitory concentration on that one? 1. And what is it for organism C? 16. Organism C is more resistant to that drug compared to organisms B and A. So knowing what kind of an infection someone has, what specific species you're trying to target, helps you decide what's the dosage or what's the concentration of the drug that I have to give somebody. Do I have to give them something that's 300 micrograms in a pill so that way when they ingest it, it gets diluted down to 16 micrograms in their bloodstream? Do you follow me? That's kind of where, like as a doctor, they have to make those decisions. They have to understand this information. At least the reasoning behind it. I'm assuming that somebody that does these, they've got like a chart that they go to when they type in what they think the person has, and then it gives them a set of information, like a chart that tells them what the concentrations are that they should be prescribing. I'm guessing. I don't know. I've never asked a doctor. Next time I go to the doctor, I think I'll ask. Strange question, I know. But that's me. I'm a science geek. I ask those kinds of things. The other way to test a drug is one that you are familiar with. We've looked at this in several different ways. It's called the Kirby-Bauer diffusion method. So we've looked at a worksheet when we looked at chemical control. Same idea with the drugs. This is used to qualitatively determine the susceptibility of a given organism to various antimicrobial drugs. So we know that a clear zone of inhibition around a microbial disc reflects in part a degree of susceptibility of an organism. So if I'm looking at that culture dish at the top of the screen, you can see if there is just one type of bacteria on that plate, each of those discs represents a different drug. Would you say that there are 2 discs that are probably more effective against that bacteria than the rest of them? Would you say the one at about 12 o'clock and the one at about 6:30, 7 o'clock? If I can use those as descriptive terms on there. Okay. So there have been special charts that have been prepared to correlate the size of the zone of inhibition. Based on the size of the zone, the organisms can be described as either susceptible, intermediate, or resistant. Okay, so I'm looking at those 2 discs on the left-hand side close to 9 o'clock, and I would say that those 2, the top one especially, it would definitely be resistant to that drug. The one below it, probably I would put in the resistant range. It's not destroying very much. So there's a correlation of the minimum inhibitory concentration and the zone size in this Kirby-Bauer disc diffusion method, along with the determination of achievable blood levels of each drug, right? If somebody takes an antimicrobial drug, we have to know how well it diffuses into the bloodstream, because it's not going to go in at the exact concentration as it was in the pill or the, I don't know, the liquid, you know, like you give little babies the little kind of syringe full of the liquid of amoxicillin, and they have to drink it down. They usually don't give babies pills. So if you have a child and they end up with an ear infection, you got to give them the little cup. Well, if they can't drink out of a cup yet, kind of interesting. Little preview into future life for some of you. All right. If you're thinking of having kids, that is. So, resistance to drugs. So drug resistance of microbes limits the usefulness, obviously, of the known antimicrobials that we have. As these drugs are increasingly used and misused, the strains that are resistant to their effects have a selective advantage over their sensitive counterparts. For example, initially, less than 3% of staph strains were resistant to penicillin G. However, because we've relied on penicillin G so much, we have eliminated a lot of those sensitive strains so that now 90% or more are now resistant. That is a huge amount. Antimicrobial resistance is a matter of great concern to health professionals, obviously, because of the impact on the cost, complications, and outcomes of treatment. It makes me concerned as a taxpayer, and when I'm at work and I'm looking at my insurance and the premium I have to pay for insurance, our premiums keep going up. Health insurance costs more and more because the drugs are starting to cost more and more, and we're starting to see more infections, and the money's got to come from somewhere. Somebody's got to pay for it. So understanding the mechanisms and spread of antimicrobial resistance is kind of an important step at this point in curtailing that problem. So remember that there are several different mechanisms of drug resistance. So some mechanisms can include drug inactivating enzymes. So perhaps the cell has a plasmid that codes for the production of an enzyme that makes the drug ineffective. That's one way the cell might be able to survive. Perhaps it's the alteration in the target molecule within the cell. So maybe the production of whatever that target is in the cell, not necessarily an enzyme, but another structure, maybe it is slightly different in those cells that have resistance. It could be that the cell is coded that it doesn't take up as much of the drug. So even though the drug is present in the concentration that's supposed to work, that targeted cell doesn't take it in, and therefore it's ineffective. And in some cases, some resistant cells have the ability to eliminate the drug entirely, break it down. So those are the different ways that a cell could be resistant. Whew. So this is where we've kind of studied some of this before. Think about transduction, transformation, and conjugation. Keep those mechanisms of gene transfer in your mind. But we've also already talked about the fact that spontaneous mutation might be one way that they could become resistant, or they could acquire new genes. And those new genes might be through one of those mechanisms of transfer. Spontaneous mutation from one generation to the next is called vertical evolution. Take your hand and make like a vertical line in the air. Think parent cell to daughter cell or offspring. So that's vertical evolution or vertical transmission. In contrast, if there's gene transfer, like transduction, transformation, conjugation, that's called horizontal evolution because that's going from one cell of the population to another. So take your hand and do a horizontal line, and that is those mechanisms of transfer that we've already studied. So the most common way is conjugation, and that's through those transfer of those R plasmids. We've already studied that. In addition, transposons play an important role in the transfer of antibiotic-resistant genes. Excuse me. Questions? That one looks like a big slide, but it's really not, because we've already talked about a lot of it. So, innate or intrinsic resistance means that those antibiotic targets simply are missing from the bacterium. For example, Mycobacteria that don't have a cell wall are penicillin resistant. Okay? They're penicillin resistant. Penicillin doesn't work against peptidoglycan if peptidoglycan's not there. Alternatively, there exist a number of non-intrinsic or acquired mechanisms that can lead to resistance. So acquired resistance can occur by alteration of the target by chromosomal mutation.
Can you go back?
Yeah.
Sure. If I can do that up here, upside down. Wow, if I look over here, that'll work. There, is that what you need? So we're going back right now. There, so that way we have time to write. I apologize. If you're watching the video, you can just move forward while we wait.
We probably need to break too.
But they have the benefit of pausing the video to finish writing. You can't pause me, I just have to stop talking.
True.
Don't you wish you had a little remote or a mute button for me that you could just turn me off whenever you wanted to? Are we good? Sorry for the long pause. Take a breather, shake out your hand if you need to. Depends on what you're doing. Nice. Okay. We're going forward. So, I already talked about chromosomal mutation, or the acquisition of those plasmid encoding resistance genes. Those are plasmids. So this is just really showing you an image, a diagram of those different methods that we just talked about on the previous slide. Okay. All right. I believe we're nearing the end here, yes? So, some examples of emerging antimicrobial resistance. We will be taking a look at some of these examples later in the semester. So enterococci, these are going to be bacteria that attack the gastrointestinal tract. Not very fun to get because you get diarrhea and vomiting with it. But these are the common cause of nosocomial infections, those infections that you get from going into the hospital, where you go in because of one reason and then you leave with an infection that's entirely different. Some of them are resistant to the beta-lactam antibiotics like penicillin. Some of them, like vancomycin-resistant enterococci or VRE, are even resistant to vancomycin, which is actually a fairly strong antibiotic. Staph aureus from your skin is a major cause of nosocomial infection, and I shouldn't say is becoming resistant to antimicrobials. It is, at this point, there's high resistance to a lot of antimicrobial drugs. So methicillin-resistant Staph aureus, or MRSA, is one example. Vancomycin intermediate Staph aureus, or what's called VISA. And then Streptococcus pneumoniae. Some strains we're finding are starting to become resistant to penicillin. And then Mycobacterium tuberculosis. There are strains that are now resistant to two of those favored peers drugs that we talked about, isoniazid and rifampin. And so they're called, those strains are called multiple drug resistant Mycobacterium tuberculosis, or MDR-TB, like how they give an acronym to everything. Scary, especially if for some reason you end up being exposed to any of these. Now, why is it important for us as regular humans in society to maintain the course of taking an antimicrobial treatment? Why? Well, you might think it's working, you might feel better, but that doesn't mean that the bacteria are entirely gone, right? Isn't that kind of like the important thing that we want to have happen in an infection? So you think about when an individual starts to take an antibiotic, and think about when you've had to take one. Sometimes it's 5 days, 7 days, 8 days, 10 days. Usually it's not longer than that, but when you start to take it, you might have a large amount of intermediate organisms and probably equal amounts of highly sensitive and highly resistant organisms. Usually after day 3, the antimicrobial impacts a majority of those intermediate organisms, not all, but a majority, will decrease the sensitive strains and maybe not impact very many of those highly resistant cells. By day 6, we see a major decrease again in the intermediate organisms. Not a whole lot of impact on the resistant strains, but even fewer of the sensitive. And then when we get to day 10, what should happen if the antibiotic treatment is finished is you get even fewer of those sensitive, and hopefully most of them have been destroyed, as well as the intermediate. And there's probably more cells that are resistant present in the population, but at that point the number of cells present are so low in number that your immune system can fight them off on its own. Now, what happens if after day 6 on a 10-day cycle you stop taking those antibiotics? That's where the issue occurs. By day 6, after 5 days, people usually feel significantly better. They don't have a sore throat, the cough is gone, the mucus is gone, they're not running a fever anymore, and they're feeling pretty much like they're back to normal, so they just stop taking it. Well, the issue then becomes when you look at where you leave off on day 6, you've got quite a few of just the resistant organisms left compared to the sensitive and intermediate. So then you stop taking the drugs, and guess what? Those resistant strains, because there are so many left, begin to reproduce, and then you get a really high number of just resistant organisms present. And that's when things get really dangerous. Then you have to start to take those drugs that have a low therapeutic index and are actually more toxic to you and dangerous to you with more side effects. So if you walk away from this unit with really one key understanding, I would hope that it's your understanding of how an antibiotic or antimicrobial works, so that way you follow all of the instructions by your doctor. So in order to slow the emergence and spread of antimicrobial resistance, it's the responsibilities of physicians and other healthcare workers. So they should be prescribing antibiotics if appropriate and required. If somebody's got a viral infection, giving them an antibiotic that only attacks bacteria isn't going to help the individual, but it is going to impact the flora or biota that's present in that individual. Obviously, it's the responsibility of the patients to follow the instructions. It's going to be the importance of being educated in the public about the appropriateness and limitations of antibiotics, as well as the fact that there are global impacts on the use of these antimicrobials. So in developing countries, antimicrobial drugs are available on a non-prescription basis. You can walk into the local drugstore in Mexico and you can buy penicillin off the shelf. You can buy a lot of antimicrobials right there in the drugstore. Well, so they're not controlled. So people that don't know any better can go in and purchase them and use them, and maybe not even use them appropriately, which then impacts the population of bacteria in their bodies. So another worldwide concern is the use of antimicrobial drugs in animal feeds. If you give antimicrobials to cattle, and then people end up drinking the milk, those antimicrobials are in the milk. You're drinking antimicrobial drugs, not in super high concentration, obviously, but you're still drinking them, you're still ingesting them. You have a steak, right? Maybe they're putting antimicrobials in the grains of the chicken because they're keeping the chickens in cages and in close proximity, and they're trying to prevent the chickens from getting infections so that way they can crank out lots of chickens on the farm to make more money. Well, then you eat the chicken, you're eating the antimicrobials as well. So it's something that you probably studied in ecology and biology, in which there's kind of this build-up effect as you follow the food chain. So the more you ingest, the more it's in your body, the more you're exposed to it, the more it impacts your normal flora without even knowing it. Oops. Now I'm pushing buttons. What just happened there? Good on you guys, but not on this one. Well, that's good because we're done. Thanks for being patient.